Hi everyone, I am Anna. I'm a PhD student from the Leuven Brain Institute at KU Leuven, Belgium, and today I will present my study that I'm currently working on, which takes a closer look at rat vision and using rats as a model for vision research. Studies on visual recognition in humans and monkeys have shown that two specific features are important. On the one hand, shape features have been proven to play a role in visual recognition, and on the other hand, we have contrast features. A simple shape feature is whether a line or edge is straight or curved, or whether, for example, an object is elongated or squared. An example of a simple contrast feature is that the top of an object is lighter than the bottom. Some rodent studies have suggested that shape features also play an important role in rat vision, whereas other studies highlight the importance of contrast features. My study will focus on this part specifically, and we will use stimuli that have been used in human and monkey studies to investigate whether rats use contrast features in face categorization. Contrast features in face categorization have been incorporated in a simple computational model for face detection. This model constructs a face template that is invariant to illumination changes. A face is detected in this model if the 12 conditions on the right are met. These conditions correspond to pairwise contrast relationships between facial regions. From previous in-lab research, we know that rats are capable of performing a face discrimination task. We know that contrast features play an important role in primate face recognition, and therefore we want to use stimuli that Ohayon and colleagues have used to investigate whether rats use contrast features to discriminate between faces and non-faces. I will call faces from now on targets and non-faces distractors. We retrieved all stimuli from Ohayon and colleagues, but for our study, we did not need all stimuli. So we chose our stimuli based on two measurements, template similarity and pixel dissimilarity. The template similarity categorizes the stimuli based on how many correct contrast features they have by checking how many of the 12 conditions are met. Within each category of the template similarity, we then calculated the pixel dissimilarity by comparing each pair of stimuli pixel-wise. When choosing our stimuli, we ensure to pick stimuli that are as dissimilar from each other as possible. We focus on a high dissimilarity within our training set and also a high dissimilarity of the test set relative to the training set. Taking these two measurements into account, we selected these stimuli for our training and testing sessions. We have a different number of targets and distractors in our testing set because we want to test 6 different conditions that compare the different number of correct contrast features between targets and distractors. If rats use contrast features in a face discrimination task, we expect their performance to be higher the larger the difference is between correct number of features between targets and distractors. In this experiment, we used long Evans rats in a touchscreen setup, and the protocol started with a standardized shaping procedure to accustom the animals to the touchscreens. Once they have completed this, they performed the face discrimination training and testing twice. During testing, 2/3 of the trials consisted of testing stimuli, whereas 1/3 corresponded to training stimuli. Each session lasted for 60 minutes or until they performed 100 trials. To give you an idea of how many days and trials each animal performed, one animal performed 5,885 trials for both training runs together and 2,080 trials for both testing runs. Here, each bar plot represents the performance of all rats together on one of the 6 conditions that I recapped on the right. We compared the performance of the old versus the new stimuli during testing. For conditions 1, 2, and 6, we find a significant difference to chance level for the new stimuli. These results are somewhat in line with the performance that we predicted or expected. However, overall, the performance of the animals on the new stimuli is rather low, and thus the question is whether we can truly speak of generalization in this case. To take a closer look at our data, we calculated a pairwise percentage matrix where each cell indicates the average performance across all rats on that specific stimulus pair. Ideally, if we would find a strong evidence for the role of contrast features in rodent vision, we would see a clustering like this in a pairwise matrix, where each cluster or bigger square corresponds to one condition. However, if you look at our pairwise percentage matrix, this kind of clustering is not very obvious. Other interesting findings that can be seen in this matrix, however, are within condition differences of some stimuli. For example, these two targets. Although they are categorized in the same condition, namely 6 correct features, the performance of the animals on these targets are very different. The first target has even a better performance than the targets in the 12 correct features condition. However, the number of correct features might be too simple as a metric, given that features can differ in how big the differences are. We therefore decided to compare this pairwise matrix with what we call a feature strength model. This model gives weights to each correct feature based on how big the contrast is for that feature. Again, you can clearly see the within condition differences between some targets, similar as for the performances of the animals. If we then compare our pairwise matrix with this model, we get a correlation of 0.37, suggesting that this model provides an explanation for our results. A next step in our study was to investigate what different layers of a deep neural network would do when trained and tested on our stimuli. We also wanted to see whether our feature strength model is similar to the performance of the neural networks. To do so, we used AlexNet after the standard training on 1,000 categories from the large ImageNet database. We trained a linear classifier on the unit responses of single layers in this network to our 10 training stimuli, and tested how well the classifier could generalize to the test stimuli in our experiment. The findings show that the network had no problems to achieve high performances in the testing phase, even not with relatively low layers in the network. These findings did not change when we added noise to the network. When correlating the performance for single target distractor pairs between single neural network layers and the red performance, we find that earlier layers indeed show some correspondence to the contrast model, whereas the higher layers show more similarities to the red performances. Taking the results of our behavioral study and this computational part into account, it seems that the animals use contrast features, but that there is something more to their vision which we cannot explain with that model, and that is captured by later layers in a DNN model. Thank you for watching my video on my study, and I would like to take this opportunity to thank our lab technicians Emily van Emten and Louise Monen, who helped us in executing this experiment. If you have any questions, feel free to drop them here.